Evolutionary developmental biology refers to the study of developmental programs and patterns from an evolutionary perspective. It seeks to understand the various influences shaping the form and nature of life on the planet. Evo-devo arose as a separate branch of science rather recently. An early sign of this occurred in 1999. Most of the synthesis in Evo-devo has been in the field of animal evolution, one reason being the presence of elegant model systems like Drosophila melanogaster, C. elegans, zebrafish and Xenopus laevis. However, in the past couple of decades, a wealth of information on plant morphology, coupled with modern molecular techniques, has helped shed light on the conserved and unique developmental patterns in the plant kingdom. Also, historical perspective. Before 1900, the origin of the term morphology is generally attributed to Johann Wolfgang von Goethe. He was of the opinion that there is an underlying fundamental organization in the diversity of flowering plants. In his book The Metamorphosis of Plants, he proposed that the Bauplan enabled us to predict the forms of plants that had not yet been discovered. Goethe also was the first to make the perceptive suggestion that flowers consist of modified leaves. In the Middle Centuries, several basic foundations of our current understanding of plant morphology were laid down. Nehemiah Grew, Marcello Malpighi, Robert Hooke, Antony van Leeuwenhoek, Wilhelm von Nageli were just some of the people who helped build knowledge on plant morphology at various levels of organization. It was the taxonomical classification of Carolus Linnaeus in the 18th century, though, that generated a firm base for the knowledge to stand on and expand. The introduction of the concept of Darwinism in contemporary scientific discourse also had an effect on the thinking on plant forms and their evolution. Wilhelm Hofmeister, one of the most brilliant botanists of his times, was the one to diverge away from the idealist way of pursuing botany. Over the course of his life, he brought an interdisciplinary outlook into botanical thinking. He came up with biophysical explanations on phenomena like phototaxis and geotaxis, and also discovered the alternation of generations in the plant life cycle. 1900 to the present, the past century witnessed a rapid progress in the study of plant anatomy. The focus shifted from the population level to more reductionist levels, while the first half of the century saw expansion in developmental knowledge at the tissue and the organ level. In the latter half, especially since the 1990s, there has also been a strong impetus on gaining molecular information. Edward Charles Jeffrey was one of the early Evo-devo researchers of the 20th century. He performed a comparative analysis of the vasculatures of living and fossil gymnosperms and came to the conclusion that the storage parenchyma has been derived from tracheids. His research focused primarily on plant anatomy in the context of phylogeny. This tradition of evolutionary analyses of plant architectures was further advanced by Catherine Esau, best known for her book, The Plant Anatomy. Her work focused on the origin and development of various tissues in different plants. Working with Vernon Cheadle, she also explained the evolutionary specialization of the phloem tissue with respect to its function. In 1959, Walter Zimmermann published a revised edition of Die Phylogenie der Pflanzen. This very comprehensive work, which has not been translated into English, has no equal in the literature. It presents plant evolution as the evolution of plant development. In this sense it is plant evolutionary developmental biology. According to Zimmermann, diversity in plant evolution occurs through various developmental processes. Three very basic processes are heterochrony, heterotopy, and heteromorphy. In the meantime, by the beginning of the latter half of the 1900s, Arabidopsis thaliana had begun to be used in some developmental studies. The first collection of Arabidopsis thaliana mutants were made around 1945. However, it formally became established as a model organism only in 1998. The recent spurt in information on various plant-related processes has largely been a result of the revolution in molecular biology. Powerful techniques like mutagenesis and complementation were made possible in Arabidopsis thaliana via generation of tDNA containing mutant lines. Recombinant plasmids, techniques like transposon tagging, etc., availability of complete physical and genetic maps, RNAi vectors, and rapid transformation protocols are some of the technologies that have significantly altered the scope of the field. Recently, there has also been a massive increase in the genome and S sequences of various non-model species, which, coupled with the bioinformatics tools existing today, generate opportunities in the field of planet.Evo-devo research. Cussard provided a detailed in-depth analysis of the history of plant morphology, including plant development and evolution, from its beginnings to the end of the 20th century. Organisms, databases and tools. The most important model systems in plant development have been Arabidopsis and maize. Maize has traditionally been the favorite of plant geneticists, while extensive resources in almost every area of plant physiology and development are available for Arabidopsis thaliana. Apart from these, rice, Antirrhinum majus, brassica, and tomato are also being used in a variety of studies. The genomes of Arabidopsis thaliana and rice have been completely sequenced, while the others are in process. It must be emphasized here that the information from these model organisms formed the basis of our developmental knowledge, while brassica has been used primarily because of its convenient location in the phylogenetic tree in the mustard family. Antirrhinum majus is a convenient system for studying leaf architecture. Rice has been traditionally used for studying responses to hormones like abscisic acid and gibberellin, as well as responses to stress. However, recently, not just the domesticated rice strain, but also the wild strains have been studied for their underlying genetic architectures. Some people have objected against extending the results of model organisms to the plant world. One argument is that the effect of gene knockouts in lab conditions wouldn't truly reflect even the same plant's response in the natural world. Also, these supposedly crucial genes might not be responsible for the evolutionary origin of that character. For these reasons, a comparative study of plant traits has been proposed as the way to go now. Since the past few years, researchers have indeed begun looking at non-model, non-conventional organisms using modern genetic tools. One example of this is the Floral Genome Project, which envisages to study the evolution of the current patterns in the genetic architecture of the flower through comparative genetic analysis, with a focus on S sequences. Like the FGP, there are several such ongoing projects that aim to find out conserved and diverse patterns in evolution of the plant shape. Express sequence tag sequences of quite a few non-model plants like sugarcane, apple, lotus, barley, cycas, coffee, to name a few, are available freely online. The CyCat Genomics project, for example, aims to understand the differences in structure and function of genes between gymnosperms and angiosperms through sampling in the order Cicadales. In the process, it intends to make available information for the study of evolution of structures like seeds, cones, and evolution of life cycle patterns. Presently, the most important sequenced genomes from an Evo-devo point of view include those of A. Thalasana, poplar, Vascomatrella patens, maize, and Chlamydomonas reinhardti. The impact of such a vast amount of information on understanding common underlying developmental mechanisms can easily be realized. Apart from Western genome sequences, several other tools like PCR, yeast-2 hybrid system, microarrays, RNA interference, SAGE-QTL mapping, etc., permit the rapid study of plant developmental patterns. Recently, cross-species hybridization has begun to be employed on microarray chips to study the conservation and divergence in mRNA expression patterns between closely related species. Techniques for analyzing this kind of data have also progressed over the past decade. We now have better models for molecular evolution, more refined analysis algorithms, and better computing power as a result of advances in computer sciences.